Hello and welcome to my presentation. So this is my case study for Farm 001B. Um, this is a series of presentations and it is a theoretical development program, and it's my project I'm doing because I want to become an expert, which I think I have become now, in CAR T-cell therapies. And I also want to explore the regulatory requirements and the scientific rationale for bringing or for developing, conceptualizing, developing, and taking through non-clinical studies, clinical development, marketing authorization, and beyond, so post-approval activities. So I want to cover the whole of the life cycle activities for these therapy products. As I said, that this is a theoretical product. They're not currently in clinical development, and they're not been approved for marketing. They're just scientific, theoretical, made up, if you like. They are based on contemporary scientific advances, as I always say. And as I say, they're here to. I'm doing this to illustrate how CAR T-cell therapies might be used to develop and treat cancers. And the main focus is going to be on hematological malignancies and solid tumors. So this is going to be what I'm going to cover in this very long presentation. I'm just going to quickly go through it. Overview of activities for this case study. There's just been some amendments that I just want to advise people of. I just want to give another recap with regards to the product description so that we're all on the same page. I'm going to talk about manufacturing process developments. I'm going to talk about GMP Insights from the EMA documents, guidance documents. I want to talk about cell banking, the cell banks that are used, and the master viral seed and the working viral seed, what they are exactly, because it's not immediately obvious unless you know about these things. I'm just going to give a very, very brief overview on viral safety evaluation. I want to talk about the European guidance on raw materials of biological origin. So this is human or animal origin. And then I'm going to talk about decentralized manufacture in the UK, because it's now been implemented in the UK. That's come into law, and you take this approach as opposed to the centralized approach. I'm going to put some more meat on the bones with regards to the non-clinical development program and the clinical program development as well, and clinical aspects. And then I'm going to go through the ICH M4Q. I'm not going to go into much detail because that's going to be in the next presentation, where I'm going to put a lot more focus on there. But I'm just going to go through it to talk about how I think it can replace my life cycle management manual that I decided to put together. Great minds think alike. So this is the amended case study, and I've just updated it in line with the recent guidelines and in line with the recent changes in regulation for pharmaceuticals products of all the different entities. So as I say, it's milestone 1 to 6. This is the second presentation for milestone 2, and the second and final presentation. But I'm going to be talking about ICH M4Q. So this is the presentation of the CTD dossier, and I'm going to use that as the basis of my discussion for the quality aspects. So manufacturing, CMC, and the quality control. In milestone 3, I will be looking at the information that should be provided in the dossiers. Milestone 4, the clinical studies. What I'm going to do is do a dummy run, as I'm going to do one for clinical trials. So investigational new drug applications and clinical trial regulation and clinical trial in the UK. I'll try and do a dummy run of this and what information should be where. And then I'm going to do a marketing authorization application. Again, that will be a dummy run. I want to go through the new variation guidelines as it's been updated, and I haven't been through it for a while, so I want to go through that and keep myself up to speed. So this is going to be an overview of the journey that I'll be taking across this series for this case study, and this flowchart outlines all of the 6 milestones and all of the activities that I expect to complete. So I'm on milestone 2, but this is the final presentation of milestone 2, and I just want to talk about some directions and some things that we need to consider for the development plan and the considerations that we need to make in terms of moving these products forward. So this is the product description for the first product, for the first program. So LAT101 is an autologous CAR T-cell therapy generated by ex vivo transduction. So it's lentiviral vectors that are transduced in the T cells. It's designed as an all-gate tandem CAR targeting CD19 and CD22. The product is engineered with a co-stimulatory domain, CD28, and includes an inducible caspase 9 safety switch. I'm really not going to introduce it via gene editing because of the complexity, and I'm going to introduce it into the expression cassette. I think it should all go. LAT101 is bispecific CAR T designed to activate T cell upon recognition of either antigen, thereby reducing the risk of antigen escape. So it's very highly unlikely or very, very rare to have underexpression of CD19 and CD22. You either get one or the other. And so this is to ensure that all of the B cells have been targeted and destroyed, or killed. So this is to reduce the risk of evasion, the tumor or the cancer cells are being evaded from the CAR T cells. It is intended for the treatment of relapsed and refractory B-cell malignancies, including diffuse large B-cell lymphoma and acute lymphoblastic leukemia. So it can be part of a treatment program. So acute lymphoblastic leukemia, the overall intention is to go for hematopoietic stem cell transplantation, and this can be the initial step to the removal of all of the cancer cells in the first instance, and then the ability to then be able to have this stem cell transplantation. This is the second product, and it is an off-the-shelf allogeneic product, LAT202. It is engineered by genome editing using the CRISPR-Cas9 system and has an AND gate bispecific targeting mechanism. And the AND gate means that both the two is bispecific, so there's 2 CARs. They both target different antigens, if you like. One of them is the aberrant glycosylated MUC1, and the other is the HER2, human epidermal growth factor receptor 2. And the product comprises of 2 distinct CARs, each with 1 co-stimulatory domain, CD28 on the MUC1 targeting CAR and 41BB on the HER2 targeting CAR. So this is CRISPR-Cas9 engineered, genome edited basically to resist the TGF-beta mediated immunosuppression. So TGF-beta cytokines interact with the TGF-beta receptors on the T cells and causes inhibition of the T cells and basically inactivation of the T cells, and this is how the tumor microenvironment is able to evade the immune system. And so what has happened is that this particular product, or these T cells, have been armored against that mediated immunosuppression, and therefore it means that the T cells are more likely to be efficacious and have that therapeutic value. In addition to that, it has the inducible caspase 9 safety switch. This is a prerequisite, in my opinion. This product is a induced pluripotent stem cell derived product, and therefore there are specific risks associated with using this product, and therefore it is a prerequisite, in my opinion, to have this inducible caspase 9 safety switch, not just with regards to the tumorigenicity, but also with regards to the cytokine storms and the potential for cytokine release syndromes and ICANS. So this inducible Cas phase 9 safety switch has been incorporated, as has been the CXCR2 chemokine receptor that enhances tumor-directed trafficking, so that the T cells can find and are directed by the gradient, the CXCR, or the CXC, I can't remember what it is, the gradient towards the tumor, so that they are attracted to, if you like, and directed to, and find out and locate the tumor. LAT202 is intended for the treatment of advanced or metastatic solid tumors co-expressing HER2 and this aberrant glycosylated MUC2, such as pancreatic, lung, and breast cancer. So it's treatment agnostic, and the AND gate ensures the T cell activation only occurs upon simultaneous recognition of both tumor-associated antigens, thereby improving tumor specificity and minimizing off-tumor toxicity. Yeah. Right, I just want to talk about some considerations relating to the manufacturing process development. Again, as I said, I've discussed this in the previous presentation, and I want to add some more meat to the bones. So let's talk about the insights from the EU guidelines that we have to consider specifically for starting materials. So these have to be, starting materials have to be carefully qualified. And the reason being is you want to build in the quality of your product. You want to design quality into your product. You don't want to be testing for quality, and therefore your starting materials have to be carefully qualified and of an appropriate standard. Now, all starting materials have to be manufactured to GMP. This is a requirement. It doesn't matter the modality. This is for advanced therapy medicinal products, but the same is true for conventional small molecule products. The QP has to sign a declaration stating that this manufacturing product has been manufactured to GMP. So all of these products have to be qualified, but it is specifically and particularly critical because of the variability and the issues relating to, or the challenges relating to these kinds of manufacture of these biological living entities. So the amount of data to be provided for each starting material would be the same as that's required for drug substance of a cell-based medicinal product or drug substance for an in vivo gene therapy medicinal product. So, you know, the level of information is critical for these kinds of ATMPs. Or is more involved, I would say, for these ATMPs, and we're focusing on cell-based medicinal products. So, you know, genome editing components are considered to be critical starting materials, and detailed information should be provided on the manufacturing process, control of materials, characterization, process development, control of critical steps, process validation, and analytical procedures and stability. So it's basically the same thing. All of your genome editing components. So in the case of this will be the CRISPR-Cas9. So these will be the plasmids that are used to manufacture the microRNA and the ribonucleoproteins that are used for the gene, that are used for these are the components that are critical for the manufacture of your product. And it's important, these are critical starting materials, because the genome editing or the gene editing is what's responsible for giving the product its characteristics. Yeah, so it is important, it is obvious, or intuitive that would be a critical starting material, because that has a direct relationship with the efficacy and the safety of these products. And where is the information to be located? I've put information for INDs and clinical trial applications, and also for marketing authorization applications. I've used this ICH-M4 CTD structure. Basically, control of materials is the current section, CTD section in INDs and in clinical trial authorization applications through the clinical trial regulation or through the UK clinical trial applications. However, for marketing authorization, likelihood is in the future we will have a different structure, and the starting materials can have their own section, and we can include as much information in a structured way, because it's much more targeting biological medicinal products. So these genome editing components, the information, these are the sections where it should be presented in the dossier. So anything that's going through cell banking has to be manufactured to GMP. Obviously, certain things like research cell banks might not be, but they do have to be tested to GLP. Your manufacturing process usually starts from where you are going to do your routine manufacture, and that could be a cell bank, and is usually a cell bank of some sort. Right. Just have some more insights. This guideline that I'm referring to, sorry, I didn't put it in the footer, but it is the guideline for genetically modified medicinal products containing genetically modified cells. So genetic modification of cells is a manufacturing step that is affected by a variety of inputs, and therefore control is critical. Yeah, and that's quite self-explanatory. Yeah, so you have to in target cells, the strategies, you have to define what those strategies are. So what you have to think about, how to improve the quality of these, design it, the quality of these into the product. So the genome editing components, in this case, the nucleases, have to be specifically designed. So your guide RNA and your PAM, your protospacer adjacent motif on your enzyme, on the enzyme that binds to the DNA, has to be specifically designed to ensure that you have this high selectivity for the sites, the location on the genome that you want to be modified or manipulated to increase on target the likelihood and the chances and frequency for on-target editing, and to reduce off-target editing. And you need to justify what steps you've taken in your application. Yeah, the protospacer adjacent motif, sorry, has to, well, you can think about designing the accuracy into that, and mainly also the guide RNA. These are the two components that are critical for selectivity, and the issues relating to mismatch between your guide RNA and the target DNA are the main sources of off-target edits. And there is some research on engineering Cas9 enzyme, specifically or especially the PAM interacting domain, or the use of orthologs of Cas enzymes from different bacteria to improve selectivity. For the time being, I'm just going to use Cas9 as my model. These are critical changes. You can't not. This has to be thoroughly validated, and you have to provide detailed descriptions of your genetic modification. Yeah, and your manufacturing process, specifically on your modification, has to be validated. Your multiplicity of infections should be kept to a minimum, but it has to be effective. You have to show that it's effective, and you get the appropriate level of gene expression and your receptors on your T cell surface. You need to address your on and off target modifications as part of your process development and characterization. So you can not always detect off-target modifications. It may not always be you may not always be able to detect it. Although we will we will be selecting a clone that has the best and the most highest purity in terms of on-target modification and the lowest in terms of off-target modification. But you need to address in your process development and characterization studies, you need to basically demonstrate on target modification and as part of your process development, as part of your development throughout your manufacturing development program. And again, you need to do risk assessments to address the potential appearance of off-target manufacture that may have occurred during your manufacture. And this is where you would have done looking at your bioinformatics and looking at your in silico assessments to see what potential off-target modification could have occurred that you perhaps might not have detected. Right, this is a slide on viral safety evaluation. So anything that goes into human beings, we have to do viral safety evaluations, and this consists of viral clearance studies. This is in line with ICH-Q5A. I'm not going to go through this guideline in detail. I have read it in detail. It took me ages, but I'm not going to go through it in detail because it will take a long time, and it's not, I'm just going to give a top level overview of the points and what it's going to be addressing. And I think I didn't really finish this, but anyway, so you need to do your viral safety evaluation. You need to account for the absence of adventitious or viruses or endogenous retroviruses. So you need to be performing tests on permissive cells or perform tests by qPCR. Yeah, so you need to be able to identify potential viral DNA or viral nucleic acids. What you really want is to confirm the absence of adventitious agents, and well, you can't really necessarily have the absence of endogenous viruses if you are using materials of human or animal origins. But these endogenous viruses what steps can in your manufacture could potentially induce them to become activated if they're not activated and they're latent. So and really, when we're talking about endogenous viruses, we're really talking about retroviruses in this particular case that are integrated into the genomes of these cells. So they may not be detected, or they may be at very, very low levels, but what are the risks of potential activation of these viruses, and what are the risks to humans in terms of the tropism of these viruses to human cells. So we have to do that risk assessment, but really what we want is to basically confirm this viral safety evaluation and studies is to confirm the absence of these not confirm, but to give confidence in and to design your manufacturing process so that you have a high degree of confidence that there are that you've addressed any potential adventitious agents or endogenous retroviruses. So it's basically an evaluation of your manufacturing process's capacity or capacity, I would say, to clear or inactivate identified viruses. And you would perform your tests of your unprocessed bulk to test for any viruses, and this will be the basis of viral clearance studies. Your viral clearance studies will be done on a scaled down model of your actual manufacturing, commercial manufacturing or clinical trial manufacturing method, because you can't introduce spiking, because spiking is one of the tests that you would use to perform. You'd need to perform in order to demonstrate inactivation or clearance, and you don't want to introduce that into your manufacturing plant. It's not good manufacturing practice to do that. So you would do your evaluation on a representative version of your manufacturing facilities. Your risk assessment. So your risk assessment is to look at your sourcing, your materials, look at your manufacturing process, your raw materials and starting materials, and to do a determination as to what kind of viruses are likely to be present and what are the impacts on humans, what are the impacts on the patient in terms of human tropism, cell tropism, sorry, and what are the likelihoods and the probability of your manufacturing process eliminating or inactivating these potential viruses. You would need to do virus clearance studies and select the relevant viruses or model viruses. So there can be specific model viruses or non-specific model viruses. So your risk assessment is a very high level and intensified activity, and you would need to include a lot of people, in a lot of stakeholders involved in that, and they would need to assess the risk to human health and the sort of infectiousness to humans, as well as the quality by design aspects, so the sourcing and the traceability of certain materials of human and animal origins. And your risk assessment will give you the actions that you need to mitigate any risks that have been identified, and then you would need to test your manufacturing process to ensure that that mitigation process, those mitigation steps will actually work. And then the other thing that you need to consider is to minimize the risk of transmitting agents causing transmissible spongiform encephalopathies of materials of animal or human origin. So that's another aspect, and that's about where you source your materials and the steps implemented to prevent the risk via transmitting agents such as prions. I want to talk about the European chapter on the requirements for risk management, if you like, for raw materials used in the manufacture of advanced therapies such as cell-based and gene therapy medicinal products. So this chapter in the European Pharmacopoeia, it focuses on the safety and the efficacy of and quality, I guess, of the final product by controlling the quality of the starting materials and the raw materials, yeah, especially and particularly those of biological origins. And this is how we do it, and this is the steps that are involved. But I just want to say that well, let's just quickly go through the steps. So you're going to look at the quality and suitability of your materials. So you have to have reasonably grade materials. It's not always easy to get them for biological entities or materials of human and animal origin. It's very difficult to control and get that consistency, can be very difficult, although we're getting better at it now. And there is an acknowledgement that research grade materials may only be available for novel products, yeah, and therefore you might not be able to get European Pharmacopoeia grade materials or standard grade materials. You might have to use research grade materials for novel technologies. And in that case, it's up to the manufacturer, the marketing authorization holder, or the sponsor or developer to make sure that they perform a thorough risk assessment, and they implement control strategies to mitigate risks associated with using these non-pharmaceutical grade materials. So I think I've talked about it here, this risk assessment and mitigation. So you have to take a risk-based approach. So identify and evaluate the risk posed by each raw material. So this is very much following on and in the same genre, if you like, of the viral evaluation. And in this particular case, we're talking about looking at all of the individual raw materials, not just from a viral perspective here, but also from their perspective in terms of the final drug product. We want to make sure that we get that consistency, batch to batch consistency that gives us confidence that we have a high quality, high therapeutic efficacy product, because the efficacy is linked to the quality. Your traceability and origin is important. You want to be able to audit, and you want to be able to understand the events that have occurred to those cells. If, say, for example, you're looking at cell lines, you want to be able to make an assessment as to whether or not any events that have occurred to those cell lines, whether they have an impact on the final drug product. And it's good also because the regulators want to review that data in terms of them making their assessment whether or not you've provided them with enough information on the control strategies. And then your testing and control, these are the regular, if you like, or the typical testing that you would need to do for critical quality attributes. So identity, purity, sterility, absence of adventitious agents such as bacteria, fungi, viruses, and mycoplasma. So your biological raw materials. So anything, animal derived components are, you know, they tend to be used because of their specific properties and characteristics. But where possible, and the use of animal derived free components, free policy encourages the use of materials that are completely free from animal and human components. And this is to reduce the risk of pathogen transmission, so prions and zoonotic viruses. Batch to batch consistency. So really, you want to make sure that, as I've stated before, your raw materials and some critical starting materials based on human and animal origins, that's where you're going to get your variability. So you want to reduce, or we want to control the quality of your raw materials in order to reduce this risk of batch to batch variation. You have to quality systems, pharmaceutical quality systems, means that you have a change control in place, and that change control may require you to submit an amendment, or it may require you to do a variation, again depending upon the impact of quality on your product. So these biological raw materials, they're very critical to the they're very critical to the quality of your product, and any changes that you make to your raw materials may require you to, well, most probably will require you to do an impact assessment. You have a change control system as part of your pharmaceutical quality systems, and you need to determine if that change has any significant impact with the relevant stakeholders on the quality. It might not always be possible to tell through analysis of data, and it might be that you may be required to generate stability data or other kind of efficacy data, such as non-clinical studies or clinical studies, that highlight the changes are significant. So you really do need to monitor all changes in your raw materials, particularly for these CAR T-cell products. I now want to talk about cell banks because I mentioned them in my previous presentation. I just want to give a little bit more background into them. So cell banking, what does it originate from? Cell banking should be manufactured. All the cells that you're going to be using for your routine manufacturing process of your drug substance and drug product should be manufactured to GMP, and it should incorporate the principles of quality by design, and they should be well characterized. Not all of them, but the master cell bank, working cell bank, and end of production cell banks, you should have some characterization activities, particularly for your master cell bank. So your research cell bank. So this is the initial um, banked version of for your production line. It's not usually not manufactured to GMP, but all the testing should be done to GLP. Um, this is the research cell bank is where we select the clone. So for our cell substrate, we select a clone from your research cell bank. So this is, and the clone is then expanded to form and pooled to form your master cell bank. Your master cell bank, this is your primary source and it's the cornerstone, and it's prepared from a single pool, so it could be a clone, and it's GMP manufactured, it's derived from your research cell bank, and it is extensively characterized. Your working cell bank, this is what's used to manufacture your products. Now, this is just, I mean, I'm going to go through another slide where we talk about master virus seed and working virus seed, but let's just talk about this for the time being. But in any case, your working cell bank is used to initiate your production line. It means that your master cell bank is protected. You don't need to do as much testing, you just do limited testing, but you do a lot more testing on your end of production. So your end of production, or your limit of in vitro cell age, this is a regulatory requirement. It is banked. It is there to demonstrate genetic stability and the expression of your genes, so phenotypical characteristics of your product. So it's there to demonstrate genetic stability and safety, and it is a regulatory requirement. And again, your viral safety evaluation has to involve these banking system, and it's a critical part of that. I just want to give a very brief overview on cell banks that are going to be used for this clinical trial. So your cell bank will be your master cell bank, and this is going to be the HEC293 T cells. And these are the parental cell lines without the lentiviral vector genome. So it's kind of, these will be characterized and they will be banked. And then you will form your master virus seed bank. Your master virus seed banks, these are cells, yeah? So they're not viruses, they're cells. And these cells, again, are characterized and banked. And they contain the transgene and the packaging integrated into the cell's genome. So they're cells, yeah, that are characterized and banked. And these cells are then expanded and used to produce the lentiviral vectors. And this is what we refer to as the working virus seed bank. So these working virus seed banks are manufactured from the producer cells, which are the expanded master virus seed banks. And they are the viral particles, and then they have to be fully characterized, and they are used to transduce the T cells. So the initial starting cellular material, which was derived from the patient in autologous manufacture, they were enriched and activated, purified, and then transduced with these lentiviral vectors. So I just wanted to give that overview. Right. The reason for this slide is because I missed out something in the last presentation. I forgot. I didn't mean to talk about it, but I just forgot. And it's relating to epigenetic profile. It's really important. So the induced pluripotent stem cells, these are reprogrammed, and it could be peripheral blood mononuclear cells, or it could even be T cells. I haven't looked at T cells, but there's a possibility. In fact, it might be better to use T cells that are reprogrammed, gene edited, and then differentiated back to the T cells. So in terms of the epigenetic profile. So it might be better to use those, but at the moment I'm just going on the theory with the PBMCs. So the reprogramming method has to be provided, You have to give that information because it is critical to the efficacy and safety of the drug. So we need to have that information, and that method needs to be validated. Then you need to you perform your gene editing. Your gene editing components could be sourced from qualified suppliers. You need to have rigorous third-party agreements in place for that. The design of the enzyme is important for accuracy, editing accuracy, and you have to provide the information that you would provide for a drug substance for these genome editing components in your presentation of your dossier. Clone selection. This clone selection is required for your cell banks, master cell bank, working cell bank, and end of production cells, and you need to talk about you need to perform your viral safety evaluation. I would say at this point, this will be the starting point. You may do your risk assessment prior to that, and that would include your donor eligibility determinations and your sourcing of your cells, your cell line, etc. So that will be part of your risk assessment, looking upstream of that. But you still, I would say that your viral safety evaluation will be at this point. And then differentiation into hematopoietic stem cells or progenitor cells, and then differentiation to your T cell drug products. And this will be brought about by cytokines and growth factors. Your T cell drug product could be formulated, could be frozen and stored, frozen for storage. It could be formulated in a specific drug product formulation. And the point that I wanted to talk about, and that I didn't go through last time, was the epigenetic profile. So the epigenetic is what determines the cell identity, and it's something that is critical. It is actually mentioned in the guidelines for investigational ATMPs, that the epigenetic profile is important. So it's not just my belief, it's something that is actually mentioned in the guidelines, because it changes the profile and the behavior of the cell, if you don't have the correct epigenetic profile, and it can seriously impact the safety and efficacy of the product. So that's something that we need to just include in our characterization studies, looking at the epigenetic profile. I don't know how we're going to do that, but in terms of whether or not it's a confirmatory test, or whether or not it's just something that is used to validate our manufacturing process. But I do think the epigenetic profile is an important critical quality attribute, in my opinion, anyway. The other thing that I did mention was the induced pluripotent stem cells. The intention is to use the PBMC, the peripheral blood mononuclear cells. However, there is some new literature citing the use of T cells and reprogramming the T cells, and then performing the genome modification, and then differentiating them back to the T cells, which is more intuitive, I think, because of the fact that your epigenetic profile is likely to be more consistent. I don't believe you can make all these changes and then it just goes back magically to what it is that you want it to. There's always going to be some residual effect that you don't bank on. The other thing is that I want to talk about here is the master cell bank and the working cell bank. The plasmids, the DNA plasmids that serve as a template, if you like, for the microRNA and the Cas9 protein. These plasmids have to be, they are going to be stored in a bank system, a cell banking system. They're only going to be used once we get the clone. Then we don't need to be doing them again. So they're not going to be used regularly, but they are going to be manufactured. The plasmids, the DNA plasmids, have to be manufactured to GMP, and they have to be stored in cell banks, and we have to provide that information in our regulatory submission. So the plasmid contains the gene sequence for the Cas9 and the guide RNA. And the bank systems are usually bacterial cells, so they're E. coli, and they are stored in these master cell banks, and these master cell banks, have to be tested extensively for identity, stability, and purity to ensure that you've got no bacterial or viral contamination. And this is a GMP requirement. Um, so therefore, the production of your microRNA or your protein, so ribonucleoprotein, must be with fully qualified and characterized DNA plasmid banks, and they have to be manufactured under the principles of good manufacturing practice. I don't know what I put here. Well, okay, let's just talk about the guidelines on the lentiviral vector. So the lentiviral vector is a starting material, yeah? So that's and it's a critical starting material. Yeah, so the EMA guidance states that for genetically modified cells, the unmodified cells, so that will be the initial cellular material, and the vectors used to effect the genetic modification are considered as starting materials. I think it should be critical starting materials, but hey, I might have got that wrong. So, starting materials for investigational cell-based. So I got this from the guideline on quality, non-clinical and clinical requirements for investigational advanced therapy medicinal products in clinical trials. And this was a recently adopted guideline, 20th of January 2025. And it says that the starting material shall mean all the materials from which the active substance is manufactured. So the active substance is the genetically modified cells, yeah? And the IMPD should include the definition of the starting materials and the principles of risk-based approach that may apply to determine the extent of quality data. So, you need to do a risk assessment on the actual starting materials to determine how much information you're going to provide. And the same level of information needed for the active substance should be provided for the critical starting material. So it's a critical starting material, and it may be provided in a dedicated section in S23 that might follow the S format, including stability data, to avoid confusion over GMP requirements. Yeah. So in the S23 section, you want to include the information. You want to include information on the critical starting materials, and that can follow the drug substance format. So all of the sections in the drug substance, manufacture, the control, the stability, etc. But it needs to be in that area because you want to make sure that it is a critical starting material, and therefore GMP requirements apply. Again, coming on from this guideline, so the starting material for investigational gene therapy medicinal products depend on the nature of the product. So even though we're referring to this as cell-based medicinal products, because it is genetically modified, some of the gene therapy medicinal product guidance apply. Yeah, the starting material for investigational gene therapy medicinal products depend on the nature of the products. They can be master bacterial or virus seeds or master cell banks. Yeah, and the plasmids used to transfect the packaging or producer cells. Yeah, so these have to be manufactured to GMP, the plasmids and the master cell bank. In the case of gene therapy ex vivo, so that's genetically modified cells, the unmodified cells, the viral and non-viral vectors, and any other nucleic acid and or protein used in the genetic modification of the cells and the components to produce them are considered to be the starting material. I hope that's clear. Right, so I just want to go through some important aspects from this guideline relating to the presentation of information, what information goes where. So this guideline is for clinical trials and it's for the IMPD dossier. and it relates to the quality, non-clinical, and clinical requirements. What information should go and into those IMPDs and where they should be. So it's just a guidance document, remember, because we have to take this risk-based approach to ensure that the information that we provide is proportionate and relevant to the type of product that we are including or that we are, developing. So this guideline on quality, non-clinical and clinical requirements for investigational advanced therapy medicinal products in clinical trials, it was adopted in or on the 1st of July 2025. Yeah, so let's just go through it. Um, First of all, I just want to go through clarify what is what and what we consider to be the active substance and why. But before we make any further um, before I go into that, I just want to just give a definition with regards to the what type of or classification this particular advanced therapy medicinal product. It's a cell-based medicinal product and it is genetically modified. So LAT 101 and LAT 202, they are cell-based medicinal products and they're genetically modified. And they're cell-based primarily because of the fact that their functional activity, their mechanism of action, is based upon cellular interactions, cellular characteristics, and that's what makes them cell-based. The genetic modification is the genetic properties that give those cells the characteristics and the functional components that they need in order to do what we want it to do. So they are cell-based ATMPs, but they also come under gene therapy products because of the fact that they undergo these genetic modifications, but primarily they are cell-based medicinal products, genetically modified cells. So cell-based ATMPs are heterogeneous with regards to origin and type of cells and the complexity of the product. So what they mean by that, it's not consistent, it's not pure 99% one component. It consists of a mixture of several components, a blend of several components, and we do have to identify what that blend is. So with regards to origin, so the initial cellular starting material is going to be heterogeneous, and the types of cells and the complexity of the product itself is also going to be quite complex and it's likely to be heterogeneous. Cells can be human, so they can be autologous or allogenic or animal origins. What we're talking about is human cells, and we're going to be talking about the autologous and the allogenic types. And they may be self-renewing stem cells, and that's not what we're doing. More committed progenitor cells or terminally differentiated cells exerting a specific defined physiological function. So this is where we are. We're more heading towards that terminal differentiation, although we will have stem cell memory T cells and central memory T cells, which do have some self-renewing and some proliferative abilities, but they are mainly, committed cells. And depending on the intended therapeutic effect, cell-based ATMP fulfill the definition of somatic cell therapy medicinal products or a tissue engineered product. So these are somatic cell therapy medicinal products, and they have a mechanism of action that is dependent upon immunogenic responses, and that's why they have this is the reason for them being somatic cell therapy medicinal products. They're not regenerative replacement and repairing in that context. They are genetically modified cells that have an immunogenic and therapeutic strategy. In addition, the cells may be genetically modified, which they are, with newly established genotypes and phenotypes. These are applicable for the intended therapeutic effect, in which case the gene therapy definition takes precedent. So the gene therapy medicinal products generally consist of a vector or delivery formulation system containing a genetic construct engineered to express a specific transgene for the regulation, repair, replacement, addition, or deletion of a genetic sequence. So this is incorporated in the product, but it's not the main mechanism. Well, the therapeutic transgene is what gives the cells their therapeutic ability. Yeah, but the primary mechanism of action is this functional cellular action. By using such gene therapy constructs in vivo, which is different, where ours is, genetic regulation or genetic modification of somatic cells can be achieved in situ. So that's a different scenario to LAT101 or LAT202. A gene therapy vector may also be used for ex vivo modification of cells or bacteria, which is our case. Right. So for a product consisting of genetically modified cells, the sections on cell-based products apply to the cellular components, and the sections of gene therapy products should be taken into account. So it means that you should take both um, sets of information into consideration because they fulfill both requirements. So all of these products, with regard to a risk-based approach, all of these products are unique. They have their own specific therapeutic strategy, and they have their own risks associated with it. So the extent of information has to be proportionate and has to be applicable. It has to be applicable to the type of advanced therapy medicinal product that we're dealing with. And this is what is meant by this risk-based approach. Not all of the information that is typically put into conventional medicine IMPD dossier will be applicable for the ATMP or this cell-based medicinal product. However, some information should be provided that may not necessarily be reflected in the traditional sense of the CTD dossier, and therefore this is the purpose of this risk-based approach, that we have to look at the information that we're providing, and it has to be proportionate, and the extent of information has to deliver with regards to providing the documented information for assessment on quality, safety, and efficacy. Yeah, and that's what this risk-based approach is about. I'm not going to go through the legal basis because it's not the scope of this presentation, but I do want to talk about the quality documentation. Investigational ATMPs should be produced in accordance with guidelines on good manufacturing practice specific to advanced therapy medicinal products. So anything that's going into a clinical into humans must be manufactured to GMP, and this specific type of product has its own specific good manufacturing practice to which you should manufacture to, and its own specific requirements. The important part about this is the data on the quality aspects of the investigational ATMP should be presented in a logical structure. So they're not really necessarily forcing you. There's a saying, it should be presented in a logical structure. And really, that does ideally mean that it should really be according to common technical document. But you can you can deviate if you can justify that. But in such situations, I would get scientific advice or advice from the regulators that the approach that you're taking is acceptable to them. So it should be in the common technical document format, such as that of Module 3. The data submitted in this module should be consistent with and complement other parts of clinical trial submission. So what they mean by that is that any information that you provide, say for example in the module in the quality section, should be consistent with, say for example, information that you're providing in the clinical trial package. So for example, the manufacturing authorization should be applicable to the manufacturing sites that are listed in the IMPD quality section. The labeling should be consistent with say for example, the storage conditions should be consistent with the information that's provided in your dossier. Um, you know, it should be consistent and it should complement other parts of the clinical trial submission package. Um, as usual with most dossiers, it's divided into an active substance and a finished product section. But because of the nature of these products, it's not necessarily always clear at what point the active substance stops being an active substance and is now the finished product. So you have to define that. You have to define as the sponsor or as the developer, you have to define and justify your definition as to what is the active substance, what are the starting materials, and what is the finished product. And you have to give your justification. So here it says the active substance, any intermediates, and the finished product should be identified if possible. If it's a continuous manufacturing where you have your starting material as your initial cellular product and your endpoint is your drug product, finished drug product, as can be the case for modular manufacture, then it may be challenging being able to say this is the point at which we're saying is the active substance because it's a continuous manufacturing process. So they give you that flexibility, and this is what we talk about when we're defining the risk-based approach. Um, so that would be a closed system where we have a continuous manufacturing process with the initial cellular starting material, that's the apheresis or leukapheresis product. And we get out of that a few weeks later the finished product, which should be the product that is going to be infused into the patient. um, Yeah, and basically in this section they're stating that the applicant is just you can choose where you want to start as your starting active substance and where you want to finish as your finished product. Because if it's a continuous process, then it is difficult to define at which point, if your active substance moves straight into finished product manufacture, it's difficult to define it. You don't have a separate batch per se because it's all the same process, and therefore it's up to us, the applicant, to choose where to provide the information in the active substance or finished product specs sections. Yeah, this section, it talks about basically ensuring that we um, update the information to reflect the current knowledge. So as we're developing and we're evolving from exploratory to confirmatory clinical trials, we should be doing this. Now, for these types of products, we're very rarely going to have exploratory clinical trials and then confirmatory clinical trials as separate. The nature of these products are that they have certain unique risks associated with them, and those unique risks means that um, they you can't really give this to healthy subjects, so you're not going to really be doing exploratory safety and tolerability studies as separate to efficacy studies. They're all going to be part of the same thing, phase 1 and phase 2. And so you would be expected to provide these drugs to patients who would be expecting to gain some kind of clinical effect, therapeutic effect. So still, nevertheless, you are still expected to ensure that the data that you update your dossiers with substantial modifications to ensure that you have the most up-to-date information available in terms of science, in terms of the quality requirements, in terms of the safety, and in terms of efficacy. In regards to quality, additional removal of parameters and modification of acceptance limits or analytical methods may be necessary. But in all cases, only methods that are confirmed to be suitable for the intended use should be used. I don't know what that's about. It's interesting that they talk about confirmatory clinical trials and exploratory for this ATMP because of the nature of most ATMPs. um, you wouldn't really be thinking about, you wouldn't really be thinking about doing separating those out. But then I guess as I say, the type of ATMPs that are out there are so diverse that it's possible that there may probably at some foreseeable point, or may have already occurred. This is probably required where you will have to have that separation. But from my perspective, when I think about ATMPs, I think about the fact that their changes are usually well, they have unique risks about them, and that they are usually what's the word I'm looking for? Usually permanent, and there are some potential long-term delayed effects that, in the case of genetic modification, indicate that in those cases you could be looking at oncogenesis. So you wouldn't really, in my opinion, be doing not confirmatory, exploratory studies in humans, and this guideline is really for humans. It's clinical trials and clinical use, yeah, so that's humans. But anyway, I might have misunderstood that. Right, so let's just define what the active substance is. The active substance of a cell-based investigational ATMP is composed of the manipulated or non-manipulated cells and or tissues, and additional substances, scaffolds, matrices, devices, biomaterials, biomolecules, and other components as applicable. And it's provided in those information should be provided in S23. Yeah, so your active substance, this is this of a cell-based, is composed of your manipulated or non-manipulated cells. So the active substance of a gene therapy investigational ATMP based on gene transfer method in vivo, so that's different, that's where you're transferring, the genetic transfer is occurring inside the human, inside the patient. It's composed of the recombinant nucleic acid, yeah, because that's the recombinant nucleic acid is delivered in vivo, and where relevant, the vector used to deliver it, yeah. In the case of in vivo genome editing approaches, active substance normally comprises the tools used for the intended genome editing. So this is where the gene transfer occurs in vivo. Where the gene transfer occurs in vivo, the active substance is the actual components that are required or relevant for that gene transfer. And it can be diverse as nucleic acid, DNA, and or RNA, protein, a ribonucleoprotein, that's a complex, or viral or non-viral vectors used to deliver them. So in the case of ex vivo genetically modified cells, the active substance is composed of the modified cells, yeah? So this is what we are dealing with here, and this is what the active substance is. The initial cell population, the viral and non-viral vectors, and any other nucleic acid or proteins used in the genetic modifications of the cells are considered starting materials. Yeah, so the active substance is genetically modified cells, and anything, the initial cellular samples, the viral vectors, or not in our case, or non-viral vectors, or any other nucleic acid or proteins used. This is the Cas9 complex with the guide RNA. Proteins used in the genetic modification of cells are considered as starting materials. Recombinant proteins and recombinant messenger RNAs. I think I used to refer to this as microRNAs. Many apologies, it's messenger RNAs. And the components to produce them are also considered to be starting materials, and they should be provided in S23. So in the control of materials section, that's where they should be provided. Of note, if the investigational ATMP contains additional biological, biotechnological components other than cells, reference to an active substance master file or certificate of suitability CP of the EDQM is neither acceptable or applicable. Yeah. And the only time that you can use that is for TSC certificates. So TSC CEPs you can use them for. So these are the information that should be provided. I'll go through that in another presentation. So this is the general information. So that consists of the naming, how you intend to name it, the the procedures for naming these types of products, because these types of products follow a specific format. The structure, that should be given information on the diagrammatic information on the cells, you know, just to illustrate how they work and what they consist of, as well as a genetic or diagrammatic information of the, or illustrative, for illustrative purposes, the nucleic acid, the gene transfer gene. And this is really talking about properties of the product. So that would be the biological activity, the mechanism of action, and the sorts of things that we would be looking at measuring. The manufacturer's description of manufacturing process and process control. So this would be your flow chart that will give your input, your manufacturing process, and your own process control testing or process parameters. I mean, I will go through this in another presentation. These are the different types of should be, oh right, I think I've talked about this, a clear definition of what the active substance is, the use of cell banks, whether or not their cell banks are being used, if so, why, if not, what the IMPD should contain. The material should be obtained in a compliance with good tissue practice, and this information should be provided in the dossier. Control of materials, so this will be the S23. The information should be separated according to the type of materials and the level and the extent of information to be provided. Raw materials are, you know, not expected to be in the actual finished product. However, you do need to provide them because of the risk of transmissible contamination, such as other cellular products, such as viral and bacterial agents and mycoplasma. So you do need to provide this information, and the extent of information should be provided according to European Pharmacopoeia, and this is the reference 5212, and this talks about really about the quality of material, the documentation, the traceability of the material. The A2, you might put this information in A2, which is adventitious viral safety evaluation and validation, which is required. You are required to validate any aseptic procedure that's going into human beings. And this information is in A2, and part of your risk-based approach or your risk assessment will be looking at the input and contact materials, and some of the input and contact materials will be your raw materials. Your starting materials, as we discussed, they have to be manufactured to GMP. You may, depending upon your risk-based approach, you will have to provide a high level of information, and it would follow the S format. It will be in S23, but you would put all the information that you would require in a 32S into that section for each of the starting materials where it is relevant. Your starting materials can be one of three things. It could be a single primary cell isolate, yeah, where you haven't gone through any number of cell passages or cell culturing. It could be primary cells that have been cultured for a few passages and they're just referred to as cell stocks, or it could be a well-defined two-tier cell banking system, such as a master cell bank and a working cell bank. So you have to provide all of this information. It has to be provided in accordance with it has to be manufactured in accordance with GMP, and the level of information and the extent of data is probably going to be quite significant due to the risks associated with these particular products. Yeah, and the same applies for GTMP. Obviously, this will be for lentiviral vectors, and for the donor materials for the Cas9 guide RNA and the DNA donor template, the same level of information should be applied. we have to define what the starting materials are. We have to justify why they are starting materials, why we consider them to be starting materials, and we will be quoting this guideline. And the level of information that we're going to be providing is risk-based. And the reason being is that the therapeutic function is not just a consequence of the start, well, is usually a consequence of the starting material. The starting material being the initial cellular starting materials, the cells, and that actually have that functional capacity to bind with certain antigens to kill the targeted cells. So these are the initial cellular starting material, but the genetic modification, the genetic components that are required in order to genetically modify those cells in order for them to identify the target antigen to have their therapeutic effect. Um, Virus seed banks. This is something that's important as well because you want to use that structural components. So these are for, and then your control of critical steps and intermediate. So this is part of your control strategy. It should be in your overall control strategy methodology. You would probably cross-reference it to S22. For intermediates, it's a good thing to keep the data on that because you'll be able to show that you're manufacturing of consistent quality by making those comparisons with regards to the data that's generated. So your testing of your intermediates should be collated and collected as a way of demonstrating manufacturing consistency and as a way of demonstrating comparability as well if you make changes to your manufacturing method. Process evaluation and validation. Aseptic manufacturing has to be validated. You have to validate them, but for investigator and ATMPs that are going to be used in clinical trials, you don't have to do a full validation. You just need to do a process evaluation to show that you have specifically characterized your manufacturing process, that this manufacturing process can consistently manufacture to a required quality, and that the product is manufactured in alignment to or according to the principles of good manufacturing process for ATMPs. What else was I going to say? Yeah. Manufacturing process development, as you manufacture your product, you are going to make changes, process improvements. You're going to need to have different suppliers in order to have that continuity of supply of raw materials or of even certain starting materials. And so you're going to be making changes and optimizing your manufacturing process, and you need to track that. You need to show that tracking the different versions in your in your dossier. You will be required to do comparability exercises. The extent of data that's going to be required for this is going to be dependent upon the phase of development that you're at. How far down the development pathway are you? If you're earlier on, then you don't have to provide that much, but further down, you have to provide a lot, and it's going to be difficult to make changes during an initial marketing authorization application because of the complexity of these products. So you have to take all of those things into consideration. But comparability exercise is something that is to be expected because you're going to be developing methods, manufacturing methods, analytical methods, and you'll need to demonstrate that you're still able to produce the same or better optimal results, optimal quality data. And in order to do so, you need to do comparability. I was going to say something else about comparability, but I've forgotten what it was. Characterization, yeah, characterization is important. I wonder, I would have thought that characterization should really come before manufacture, but hey, I didn't set this up. But your characterization is important because you use it as the basis of your comparability studies, you use it as the basis of your stability studies, and you use it as a basis to determine your batch confirmation, your batch release data, and also to determine your critical quality attributes, which you use to then define your critical process parameters, because anything that varies or has an impact on your critical quality attributes will need to be monitored and controlled, and therefore characterization studies are a very, very important characteristics, or very, very important part of your development. Elucidation of structure and other characteristics. Um, this is again looking at your mechanism of action in more detail, and providing what's the word, orthogonal approach and orthogonal analysis. And this is really relevant for these types of products because their mechanism of action is not just one thing, it's a series of a cascade and a series of events that occur that lead to cell death. And we, where possible, we want to be able to measure or characterize or describe and measure and identify what's going on. I'm not going to go, as I said, I'm not going to go into this into too much detail because this can be another presentation. I think control of active substances, I think I've kind of said enough here. I don't really want to talk too much about this. I think it's really quite obvious. If you've got a background in CMC, you'll get this. Yeah, I think that's really all I'm going to talk about in this particular presentation, in this particular part of this this presentation. Thank you. Right. Now let's talk about the FDA guidance, because the FDA handles the viral vectors used for in vivo gene therapies as a starting material, but it's kind of integrated into the drug substance section as a whole. So they haven't got that flexibility that the EU has, where they say you can you can put it in the 32S23 section, control of materials section, as a drug substance CTD section. But the FDA they mandate a level of detail required equivalent to the drug substance, but they favor the integration of the material in the overall drug substance section of the CTD dossier. So while the lentiviral vectors is a starting material, the FDA guidance, the chemistry, manufacturing and control information for human gene therapy investigational new drug applications that was adopted in January 2020 outlines the required structure for the CMC Module 3, which is based on CTD. So the FDA does not typically recommend placing a vector in a separate dedicated 32S module. Instead, the information is integrated, as I stated, into the drug substance and the drug product sections of the cell therapy of the dossier, basically. And where they're referring to the drug product, then that might be necessarily referring to the aspects relating to the quality. So, for example, your drug product may also be checked for replication competent lentivirals, yeah, and that's come from upstream manufacture with regards to the lentiviral vectors. So that might be it, because your drug product will consist of your active substance plus any components. So, for example, any diluents or cryopreservatives, but it's the active substance is the genetically modified cells, and the genetically modified cells have been genetically modified by critical starting materials, which are the lentiviral vectors. So this is the FDA guidance document, and remember, in the United States, this is a human gene therapy investigational new drug. So it comes under this guidance. This guidance was adopted in January 2020. I think it's the latest guidance. And I just wanted to contrast the approach with regards to the dossier, the presentation of information, and with regards to the control of materials and what is what. So let's just quickly go through some things. I don't want to spend too much time on this. I just wanted to just contrast the approach that they take for the presentation of information. So the presentation of information, as you can see, if you have seen my thing, it's in line with the common technical document, yeah? And the information is the same format, but so you have your nomenclature, your general information, nomenclature, structure, and general properties. And then you have your manufacturer. So you have your manufacturer, S21, S22, your description of manufacturing process and process controls. So here you have your batch size. I think, I don't know whether I mentioned that in the other one, but obviously it's slightly different for your common technical document where I think it's not. I can't recall. But in any case, you have your batch size and your scale where you should identify what is a batch and what are your typical batch size for each manufacturing run. So a description of how you define each manufacturing run. So that's identifying what the batch is, should be submitted with an explanation of the batch numbering system. I mean, this is the case anyway for EU. Um, and you should clearly state whether any pooling or harvest or intermediates occur during manufacturing. Again, that's important because intermediates are important ways of determining or demonstrating consistency. And your manufacturing process, a description of manufacturing process should include a process flow diagram and a detailed narrative. So this is standard for CTD. Your description should clearly identify any process controls and in-process control testing or in-process testing. So for each of your unit monitoring or unit operation, you should really, if required, particularly for critical process parameters or critical process in-process testing, you need to provide that information. And operating parameters. So this is process time, temperature ranges, and cell passage numbers, pH, carbon dioxide, and dissolved oxygen and glucose levels. So these are really critical details because changing these can impact on the quality of your cells. So again, they say we acknowledge that this information may be gathered over the course of the product development and may be submitted in a stage appropriate manner. Yeah. So here they're just going through the sorts of information that will be presented. So for example, cell culture for vector production. Yeah, and they also talk about the fact that expectation of media components and cell bank qualifications are outlined in this section A2, control of materials, S23 S3 to S sorry S23 3.2.S.2.3. So the media components, yeah, that so the cell bank qualification should be outlined in control of materials of this guidance. Yeah, vector production. Um, so this is in your manufacturing section. Genetically modified cell production. We're not dealing with irradiated cells, and filling, storage, and transportation if required. And then you'll put information, you cross-reference such information with regards to container closure system. And then control of materials. So you must provide a list of all the materials used in manufacturing and a description of the quality or grade of these materials. So the risk-based approach is built into the FDA guidance. The extent of information should again be proportionate to the risk associated with the use of those materials. The information may be provided in a tabular format, includes the identity of the material, the supplier, the quality, so e.g. clinical grade, FDA license, and the source of material, specifically animal or human or insect, because obviously there are certain risks associated with that. And again, the stage at which each material is used in the manufacturing process. Again, that's something that is required for that particular section in a tabular format for the European Union as well as the FDA. And this includes information on components such as cells, cell banks, viral banking system, and reagents as described below in more detail. It also includes raw materials and equipments that come into contact with the product, such as culture bags, culture flasks, and chromatography matrices, because you can get issues with contamination, impurities. So they need to be listed there so that you can discuss them, and tubing. So you have to provide certificates of analysis, and those certificates of analysis may be provided in appendices. Adventitious agents safety evaluation A2 section of the CTD dossier. So you might want to cross-reference them. So just quickly go back up. I wonder if they've separated it out. No, they haven't. So they've just talked about reagents, which could be there. If you are using bovine material as a source of your culture, as a supplement, then there are certain risks associated with those, and therefore you have to provide data on that. And you have to demonstrate that you can trace the data and that the quality of the data is acceptable, and that you've taken steps to mitigate the risk of transmissible spongiform encephalopathy. Porcine, there's specific viruses, zoonotic viruses associated with porcine, as are there specific viruses associated with bovine as well. So these are viral specific testing in permissible cells in order to demonstrate the absence of these products, or to look at the scientific rationale with regards to the tropism to human cells. Murine or monoclonal antibodies, again, this may be used in your manufacturing process, and if so, there are specific risks associated with them. And material from human sources, again, you need to have the right documentation in place. So they can be used or may be used as media components and culture components in order to support the culture and cell growth and cell expansion of your cells. Human AB serum may be used. The cells themselves, the initial genetic cellular material, could be autologous or allogeneic. If they're autologous, then you need to describe the collection procedures, the good tissue practice, any steps with regards to enrichment which would probably occur, cryopreservation, labeling, all of these things. Whole time, you need to provide data that supports the whole time, transportation conditions to manufacturing facilities. As stated here, you're not required to make donor eligibility determination for autologous, but you are for allogeneic cells. You have to do a lot of screening and a lot of testing for allogeneic cells because of the fact that they're derived from a source that is not the same as the recipient, and so there are certain transmission of communicable diseases that need to be taken into account. The most important ones being, but not exclusive, the human immunodeficient viruses, hepatitis B virus, hepatitis C virus, etc., and the CJD variant and the variant of CJD. So all of these things have to be excluded and demonstrated as absence. Your banking systems, master cell bank, working cell bank, qualification of master cell bank, all of this information should be in S23. Do you see? So it's very, it doesn't have that flexibility with regards to where you put the information in. It's just quite clear, as it just defines it, and you follow the guidance. I think that's the point that I really wanted to make. And I think we'll stop it there. Right, and now I want to talk about decentralized manufacture, which has now been implemented in law in the UK in July, I think it's 23rd of July 2025, and is a good manufacturing approach, if you like, for CAR T cell therapies, in my opinion. Right, so this new regulatory framework for decentralized manufacture was established primarily through that regulation, so primarily through the Human Medicines regulation, and the statutory instrument number is number 87, so it's SI 2025 number 87, and it fundamentally changes the manufacture of medicinal products by moving well, in some cases, by moving the industry away from the universal, conventional, centralized factory model into a much more controlled, distributed network that prioritizes proximity to the patient. So basically, the centralized model is where you have one or two or maybe up to 5 manufacturing plants. It's not really 5, I very rarely find 5, but maybe 2 or 3 manufacturing plants that are registered, and these manufacturing plants manufacture your drug product, and therefore you then have a distribution process in place to ensure that those drugs are distributed to wherever they are worldwide, and that requires these drugs to be stable. And in the case of advanced therapy medicinal products, these drugs are not always stable, and they're not able to withstand the distribution and the problems associated with that. And this is where this decentralized manufacturing approach can be advantageous. So what is it about? So basically, it's a shift in the manufacturing location and the purpose for that manufacturing. So you have these decentralized sites, and they can be novel locations. In order to do this, in order to get this, you have to go through this mandatory designation step, and you have that designation, and then you're able to do this approach, and therefore the MHRA are going to inspect your control sites and your control sites. Well, first of all, for this designation, you need to pass, if you like, the legal tests for either point of care or modular manufacture. So you need to test that legal the legal test needs to be approved, and then you will get this designation, and the MHRA perform that assessment. And it's basically you have this centralized control site which is mentioned on your manufacturing authorization, your MIA, or your MIA IMP for your investigational medicinal products. And then you have the spoke sites, and these are the spoke sites are where the decentralized manufacturing occurs, and these spoke sites are under the control of the holder of the MIA. So this approach is applicable to clinical trials and marketing authorization in the UK. I haven't looked at it in the US at this moment in time, and I will do for later presentations. The application timetable is 60-day timetable. You can have a 90-day timetable if further information is required, or if you need a meeting to address any potential issues. Right, so decentralized manufacture process allows the medicinal product to be made at close to the location of the patient for administration purposes. Yeah, and you have this hub and spoke manufacturing model, and it encompasses 2 categories: point of care and modular manufacture, and both are defined partly by their necessity or for geographical proximity to the user. So point of care, it's basically defined by a critical link between their manufacture and the administration of the drug. So whatever the reasons related to the method of manufacture or the shelf life or the route of administration, these medicinal products can only be manufactured at the place where the product is going to be administered. Yeah, and if it can meet that criteria, then it becomes a point of care legal designation. And then you have modular manufacture. So modular manufacture refers to products where the manufacturing occurs in some self-contained modular unit, yeah? And so it doesn't necessarily strictly mean at the patient's bedside, but these units make it easier and necessary to manufacture these products, and it's really closely linked to the needs of the patient and patient, maybe for example, patient conditioning prior to receiving personal cancer treatment. So modular manufacture is crucial again for enabling faster deployment of products, but it's kind of a much more structured approach. Um, Also, it can be useful with regards to pandemic vaccines where manufacturing in regional manufacturing might be limited. So it could be crucial in those cases. I'm not really going to talk too much about um, pandemic because that's not the focus of this case study. But what we could be aiming for, I think, would be the use of modular manufacture, because I think that we would meet that criteria. And we would want to have those discussions, those early engagement discussions with the regulators through the MHRA scientific advice meeting. Right, so what's the difference between the two? What's the advantages and disadvantages? How do they both compare against one another? So point of care with regards to modular manufacture with regards to proximity to patients. So point of care is very close or near to the treatment site, or maybe even at the treatment site, whereas modular manufacture may be near the treatment site, but it's not necessarily at the treatment site. And it's usually a centralized modular facilities. Yeah, the vein to vein time would be rapid for point of care if they were able to implement this in the clinical manufacturing, clinical site, administration site. But it would be so for modular manufacture, it would be shorter than centralized manufacture approach, but it would still be longer than the point of care. The infrastructure. So you're required to have GMP clean rooms in order to manufacture these kinds of products, and you would be required to have trained operators. So that's a lot of investment. Whereas for modular manufacture, these, the facilities are already designed with controlled environments, and it's less demanding for the clinical sites to actually implement these. So it's built into the modular units. Complexity, well, for point of care, it would be fully integrated into your manufacture site, whereas for modular, there'll be some steps with regards to delivering the drug to the patient. So there will be some, it's not, I wouldn't necessarily, I think fragmented might be a bit of an overkill, but some of the steps in modular facilities to bring the drug to the patient, there would be, more. Um, Quality control. Again, because these point of care, you would need to train up the relevant site in order to ensure that they adhere to good manufacturing practice, which includes quality control. And so this might be challenging for point of care, but for modular manufacturing, this is likely to be built into their modular facilities, and so they're more likely to have better QC potential. Complexities. Well, in the UK, the centralized site is listed on the MIA, not all of the other sites, not the spoke sites. So I think it's quite straightforward. The only complexity relates to the manufacturing authorization holder that has to oversee all of the spoke sites, all of the decentralized manufacture sites. So I think that it's more complex in terms if there are a lot more sites, a lot more point of care sites. So there's more to oversee for point of care compared to modular manufacture, where you would have fewer sites, manufacturing sites, but the complexity is controlled by the MIA, the manufacturing authorization, or the license, or the MIA IMP. Logistics is going to be very simple because it's going to be manufactured very close to the patient for point of care. And for modular, it will require shipping between the sites of administration and the modular manufacture site. Investment and training, it's going to be higher investment for local infrastructure and training because they're not set up primarily for that purpose. Whereas for modular manufacture, the centralized it's centralized investment and there's less burden for your local facilities. Right. I just want to go. This is the MHRA website on Decentralized Manufacture Hub. So it gives all the information, comprehensive information on the documentation and the application process and the legal basis for this approach with regards to decentralized manufacture. So this is the page, the front page. And here it's giving you an introduction with regards to the UK's guidelines on decentralized manufacture. So point of care and modular manufacture are collectively described as decentralized manufacture. And it's giving you the justifications for each of them. So all the decentralized manufacture will be based on a hub and spoke manufacturing model, as I discussed. And the control site will be the only location named on the manufacturer's license and will manage overall aspects of manufacturing, including generation and management of decentralized manufacturer master files. So these decentralized manufacturer master files, just have a look at that. So just quickly looking at it, it's a very simple document, I think, compared to what it could be. Straightforward, intuitive. So you have the name of the product, manufacturer, and your version number. You have your UK decentralized manufacturer master file number. You have your control sites listed. And then here are the information. I think it's just the contents of what this decentralized manufacturer master file contains. So it will have all the general information, your control sites, functions, and your management. So what are your systems in place? Manufacturing site management information, and then you'll have information on your medicinal products, which is based on your CTD dossier. So the presentation of information is based upon this, so you don't have to re-prepare documents. You have your post-approval change management protocols. Because this is applicable to marketed products as well as clinical trials. So that won't be applicable to clinical trials, obviously. So this is the actual sections that are required to be completed. So you have your general information. Describe the processes by which the control site will review and amend decentralized manufacturer master file. Information on your control site. If you're decommissioning any sites, it will be there, satellite sites. And pharmacovigilance reporting has to be specific to the, you have to provide specific information, make sure that you provide the specific information relating to each site and the control site. Administrative information, manufacture and controls. You give all the information that is required for your master file, manufacturer's master file, stability, batch analysis, all the quality information, the relevant quality information, comparability protocols. I wonder whether or not you're going to have to provide information on starting materials. Doesn't actually say, but that would be interesting to know, because that's really quite critical to your quality of your product for biological products. Version history. So this is a copy of your master file. Whoops. And then this is the statutory instrument that was used to change the Human Medicines Regulation of 2012 and the Medicines for Human Use Clinical Trial Regulation 2004. So these two regulations, which originally derived from the directives, 2000, I think it's 2001/83 EC, and the clinical trial directives, clinical trial directives, which was then, obviously the UK didn't participate in the EU clinical trial regulation because it left the EU. But anyway, here's the statutory instrument that relates to this. So this is the legislation.gov.uk that has all of the statutory information, instruments, for all of the legal information on everything, not just pharmaceuticals and medicinal products. So here we have this is the general statutory instrument and the update 87. So this, I'm not going to look at this because it's not the purpose of this statutory instrument. Number 87 relates to oh, here it is. Yes, sorry. It does relate to modular manufacture and point of care. So this is the actual regulation, number 87, that relates to to the legal basis for the implementation of this this regulation. So it basically amends these two regulations, yeah, in order to account for the modular manufacture and point of care. So these are the differences between decentralized manufacture and conventional manufacturing operations, and it may create some specific issues and challenges, and therefore they've given these guidelines, and these guidelines are they're not draft, but they're going to be updated as the MHRA gains more experience. So a relatively large number of manufacturing sites, which may be geographically widely distributed within the UK, with many in novel locations. A wide and diverse array of different manufacturing techniques, some of which may not be used in conventional centralized manufacturing. A hub and spoke structure pivotal to the regulatory flexibilities of the decentralized manufacture, which relies upon a central control site as its hub to manage manufacturing activities between individual manufacturing sites, which would be the spokes, and the challenges relating to assurance of consistent manufacture of products of comparable quality across potentially a large number of different sites. So this is going to be quite a challenge here, ensuring that you oversee and you manage and you ensure that the product that's being produced is comparable between all of the sites and of the required standard. That's one of the challenges anyway with regards to biological manufacture. The likely co-location of manufacturing sites on or within premises of other organisations such as healthcare providers. So this could be near or even in hospitals, specific hospitals that are providing specific treatments, and the need to develop products and process intended for decentralized manufacture using enhanced process understanding, where surrogate measures such as critical process parameters may be required for product release. The GMP in the MHRA is the orange guide. So that has been updated to account for this. They've given us an exemplar products for both of these cases. What's this spectrum of manufacturing? Oh yeah, this is this describes the different types of manufacturing that could um, be applicable for decentralized. So centralized manufacturing is where you have the one large scale plant. Well, not one, but you have, one to maybe five. And decentralized, you have a number of different sites, potentially different sites. There are different categories. So there's modular manufacture, and then there's point of care manufacture. And modular manufacture is where you have these specific units, and point of care is where it could be in the manufacturing site, could be within some other kind of healthcare institution. There are YouTube video recordings on this, which I've seen. Here are all of the guidance documents. So you have to have a designation step. Remember I was talking about the legal test that's required. And so you have this designation. There's a designation form, application forms. Then you have your manufacturing master file, the decentralized manufacturing master file, and then the different applications. So you do your designation first, and then you do your applications for marketing authorization or clinical trials, whichever one is applicable. Let's have a look at marketing authorization. So in your application, you should include evidence of decentralized manufacturing designation. Here they're talking about real-time release testing, because obviously if one of the critical issues is regarding the stability of the product, then you would be more inclined to do real-time release testing as opposed to the confirmatory testing, and perhaps some testing after administration to the patient. And then clinical trial authorization and good clinical practice. Let's quickly look at that. Again, you reference it, you have it, you have your decentralized manufacturer designation. You have to go through a designation step. You have a drug master file. So the IMPD information requirements continue to apply. Yeah. So there's no, just because you've given the master file, it doesn't mean that you're no longer required to provide the IMPD. It still continues to be required. Batch analysis from representative numbers of DM locations should be included in P54, which is the batch analysis section for assurance compliance. And again, real-time release testing. And then we have the guideline on good manufacturing practice, because it's going to be specific to this decentralized manufacture. Yeah, again, as it's emphasizing, it's the control site that has the is where the manufacturing license is applied or listed on, not the individual hub sites. So that's a very quick overview on the information available, the guidance information and the master file, etc., that's available for decentralized. Right, so let's talk about the non-clinical development aspects and give some more meat on the bones. So, your non-clinical studies. So, there are different guidelines on this, EU guidelines and different FDA guidelines, and I've looked at most of them, not all of them, just to get an overview of the non-clinical studies and the approaches and the requirements for them. And what the objective of non-clinical studies is to demonstrate proof of principle and to define the pharmacological and toxicological effects predictive of human response and safety. So this is in the guideline on quality non-clinical and clinical aspects for genetically modified cells. Slightly different in another guideline in terms of the understanding of what proof of principle is compared to proof of concept, but both of them saying this essentially the same thing. So you're going to be looking at your pharmacokinetics. So your pharmacokinetics will, because you're going to be looking at safety pharmacology, so your pharmacokinetics will be addressing certain things like the in vivo fate. So you're going to have to look at biodistribution, and biodistribution doesn't just mean biodistribution of the actual cellular products, because even though we aim to have a highly pure and highly qualified product, you're going to get impurities, and some of those impurities may involve lentiviral vectors, and you have to do non-clinical studies to address the potential safety impacts of those viral vector residuals in your product. Engraftment relates to the fact that the survival of your biological living drug. So you administer your drug, how much of it survives, what is its what happens on survival? Does it expand? Does it actually perform the task that we want it to do? Is it efficacious? And do we get what's the word I'm looking for? Proliferation and expansion. What are the phenotypes? All of these things have to be addressed or not identified, but they have to be examined and assessed. And we're looking at stability and persistence because we want to ensure that you've got genetic stability with regards to the product itself in vivo, and you're also looking at persistence, because these products, we want them to persist, we want them to remain for a reasonable period of time, if necessary. If it's being given as a cure, we want to be able to have some handle or some understanding of the persistence. So your pharmacological studies will include your pharmacokinetics and your pharmacodynamics, as well as toxicology information, as well as information on your route of administration. But these effects should be confirmed on a cellular level for genetically modified cells. That's according to the EU guidelines on quality, non-clinical and clinical aspects of medicinal products containing genetically modified cells. Yeah. And now we're looking at toxicity. We're looking at your vectors. So I've got another slide where I've gone into more detail on this, but as I said, these products contain lentiviral vectors, and you may have residual amounts of lentiviral vectors. So you want to look at, for example, the possibility of vector mobilization. So this is an undesirable event where your therapeutic CAR gene is packed into a new viral particle and then subsequently transferred to untreated cells in the patient or in the environment. So this requires patients who have received these cells that have been successfully modified by lentiviral vectors to be that may have been modified, if you like, by the wild type parent virus, or which behaves as a helper virus. So the lentiviral vectors have been designed so that they've got this self-inactivating long terminal repeats. So this disrupts the packaging signal, and it means that the probability of robust packaging of these transgenes into other types of viruses is less likely, but it is something that we have to address. Recombination, again, this is looking at the potential to the formation of replication competent lentivirals. Again, you need to justify that these studies can be performed in relevant animal models. So you need to have in vitro studies. You will perform in vitro studies anyway as part of your development, and ex vivo and animal models. And you just want to show proof of principle, and proof of principle and sometimes proof of concept. And this guideline, the one on genetically modified cells, it does talk about the limitations with regards to your animal models, your animal species selection, and your animal disease models. And you do have to discuss the limitation with regards to the feasibility of these models to demonstrate proof of concept. I don't know whether or not it is. I mean, some of them can be in terms of extrapolation of the data. You can demonstrate proof of concept, but I think with regards to toxicology and safety pharmacology, that might be where the limitations and the challenges are. Right, the FDA guidance on preclinical assessment of investigational cellular and gene therapy products have a number of objectives, which when you're doing your definitive studies, your pivotal clinical, non-clinical studies, or your pivotal preclinical studies, this is what you want to have as your objectives. You want to be able to establish biological plausibility. You want to be able to demonstrate that the mechanism of action that you believe is occurring or should occur for the therapeutic effect does actually occur. So you want to establish that it does actually occur in a whole living organism, animal, and that's the establishment of your biological plausibility. You want to be able to determine what the biologically active dose levels are. Yeah, this is critical information that will inform the clinical trials, and you do want to ensure that you minimize the potential for adverse events. So you want to be able to find out what the levels are that will cause a biological that will give you biological activity that you require, i.e. to kill the cancer or tumor cells. So that identification of biologically active dose levels is a critical objective. The starting dose and the dose escalation schedules and dose regimen. So your clinical trials will be based upon this. So what you choose as your minimal effective dose, that's your starting dose, and the dose escalation schedules, i.e. the period of time between each dose and the escalation schedules has to be based upon what you're planning to do in your clinical trials, and your total full dosing regimen in your animals has to be based upon what you're doing on a clinical trial. So one has to, the non-clinical preclinical studies have to mimic what your plans are for your clinical trial. So this is the objective of your preclinical assessment. And again, your route of administration is important. It's one of the potential for safety issues and safety events and adverse reactions. So you need to look at the feasibility and the reasonable safety of the investigational product's clinical route of administration. Your patient eligibility criteria will be based upon, say, for example, the selectivity of your drug. How is your drug acting? What's its mechanism of action? And is it applicable for the patient? And that will be supported by your non-clinical or your preclinical studies, your animal studies. And your parameters that can guide with clinical monitoring. So, for example, your IL-6 amounts in the blood will give you an indication with regards to cytokine release syndrome, or well, not ICANS, but cytokine release syndromes, and your on-target off-tumor toxicities in terms of the distribution profile of your antigen. The the biomarkers that are present can give you an indication as to and guide you with monitoring in your clinical trials. And then again, you want to look at the risks to public health. So, for example, viral shedding and close contacts and workers, co-workers, or anybody who may even be involved in the manufacture of these products, because we did talk about point of care manufacture. So what are the potential risks to public health, to people who are responsible for administering the drug? And we have to get that under control. Right. The FDA, they make it very clear that non-clinical studies, particularly definitive preclinical studies and pivotal preclinical studies, should be protocol driven, especially when they're used to support investigational new drug applications or submissions. So you need to have in place defined objectives. You need to have in place your endpoint, and it needs to be prospective. Prospective protocol, not retrospective. So it's got to be randomized and controlled, and preferably with, if possible, blinding procedures. Your dose rationale and scheduling should be aligned with your clinical trial plans. So as I said, your non-clinical preclinical studies have to mimic what you're planning to do in your clinical trials. You have to be statistically powered studies. So you have to have a statistical analysis plan in place, and your statistical specifications prospectively, not retrospectively. You have to have your statistical analysis plan in place. And again, you're statistically powered. You have to have just a certain number of samples available for or numbers of animals available to ensure that it is statistically relevant, and you have to have a p-value. And then your ethical considerations as well is important. So I haven't put that here. Your ethical considerations, if possible, the protocol requires you to consider the three R principles. Now, it does say, when possible, the investigation of cell gene therapy products that will be administered to patients should be used, the investigational product itself should be used in your definitive or your pivotal preclinical studies. However, it's not always biologically relevant to do that, because if you're going to have a humanized or human-derived product, it's going to most likely produce an immunogenic response. And other relevant approaches can be acceptable, and you do need to discuss that with the regulators. Yeah. Again, this is on the same vein, and that is that it's not going to always be possible to use the human-derived product, and you will have to consider analogs. It may be appropriate. You need to go and get advice from the FDA, and this is due to species-specific nature of clinical trial products. Some cell and gene products intended for clinical administration in animals may not be informative. You're not going to get. Maybe you might get some information, but if you have an immunogenic response, you're not going to get. That's not going to give you any information, so it's not going to be informative, and therefore the testing of an analogous product may be a suitable approach. The other thing is that you need to ensure that your animal species is biologically relevant. Yeah, so it's a biologically relevant animal, and you need to justify that. So you need to perform an assessment, and you need to provide a summary to show that this is the case. that it's a, you know, so an assessment and summary is to be provided that supports the relevancy of each animal species to support the potential clinical trial. Yeah, so that's really important, and one of the requirements is that you need to demonstrate that the animal species that has been selected is biologically relevant, and you would really need to get the not the approval but the insight from the FDA prior to performing and carrying out these prospective clinical trials, and that's why you need to have your protocol in place, and that protocol would really be reviewed by the regulators in their scientific advice meetings. Again, you need to select your animal model for disease, and you can't really use healthy animals because it's not going to give you the information you require. It's not ethical either, but because of the mechanism of actions for these particular drugs, you know, you need to look at and the issues with persistence, and you need to look at disease models that will give you more information on how these drugs are going to play out in a diseased human being. And the other thing is that as we said, there are limitations with regards to animal species and animal disease models, and you need to provide that information and discuss the limitations of the potential animal models. So that's a requirement. I also wanted to make a point that before you do any of your definitive preclinical studies, the FDA do recommend that you do conduct some in vitro studies and some in vivo pilot studies to, you know, establish your biological relevance of the specific animal species to the investigational product. So all of these things, because you're not going to really, you may not really know that in advance. You need to do some preclinical studies and some you know, some, I don't know, pilot studies of some sort to determine what is the best animal species, what is the best disease model, and then design your prospective protocol around that, and then take that to the regulators for review. You know, due to like the features of these products, you know, with regards to prolonged duration and intended product effect, and the complex mechanism of action, yeah, the diseased animal models of the disease is preferable to healthy animals. Yeah, I've already discussed that. And you know, animal models of diseases help us to understand the risk-benefit ratio as well. So if you're using healthy animals, you can't really see what the benefit is compared to the risks associated to it. But if you're using diseased animals or animal disease models, you can understand the risks and the benefits of the investigational cell and gene therapy product. Again, as I said previously, in the objectives, the disease model helps us to identify the specific risk biomarkers that may be applicable for monitoring in your clinical trials. So it's, you know, there's a reason for using these animal models of diseases compared to using healthy animals. Right, so these are the examples, if you like, of preclinical models, animal models that can be used for CAR T cell therapy. I think I should have just put examples, but anyway, I'm really talking about, I've used the term preclinical here because I'm really talking about the protocoled definitive or pivotal non-clinical studies that precede your clinical trials in human beings. Yeah, so that's what I mean when I talk about preclinical in this particular context. I still mean non-clinical stuff. So it's not related to clinical trials, but these are the studies that are required to support the clinical trials, and these are the animal models that are available to us. So the first one is where we have this immunodeficient mouse, so it could be an NSG mouse, yeah, and you have xenografts of the tumor and the human CAR T cells, and this is to demonstrate they're co-injected into the mouse, and because the mouse is immunodeficient, it doesn't have its own immune system, you're not going to get that rejection. And this is just to demonstrate the proof of concept or the proof of principle. So you could use, instead of using I don't know, cell lines that are derived from, you know, traditional cell lines, you could use patient-derived xenografts instead of immortalized cell lines. But this is one approach, and this is a basic approach where you would use to demonstrate proof of concept or proof of principle. And then there's the humanized mouse model. So this is an immunodeficient mouse. It has xenografts of tumor and a fully functional human immune cells. So you can get a much more realistic understanding of toxicity, because if it's got the human immune cells, you're going to get that interaction between the T cells and the immune cells. So you know your T cells, when they're activated, they express they do express CD137, which on their surface, which interacts with other immune cells and is responsible for persistence. In addition, when these T cells are activated, they also interact with other immune cells, which are responsible for then secreting interleukin-6, which is responsible for implicated in cytokine release syndrome. So, you know, this gives you a much more realistic understanding of the toxicities, human-specific toxicities, and it gives you a better understanding with regards to persistence. And variations on that is that you can use human stem cells to engraft mice with human CD34 plus hematopoietic stem cells for a more complete long-term human immune system. So the humanized mouse model is a good model. It gives you information on human-specific toxicity. It gives you information on persistence, both of which are important parameters that need to be, measured in animals, in humans actually. And cytokine release syndrome and ICANS are specific adverse reactions from these types of drugs. Now, the next one is syngeneic models. So these are immunocompetent mouse, where we have a specific analog of the drug. So you have these mouse CAR T cells that have been transduced with lentiviral vectors containing a specific mouse transgene. Yeah, and here we're looking at genetically engineering certain mice to spontaneously develop tumor microenvironments, and then we can understand the interaction between our drug and the tumor microenvironment. So this kind of setup is really to understand how the CAR T cell functions against an intact immunosuppressive environment that is, that is of the tumor microenvironment, and particularly useful for our studies because we are armoring LAT202 to deal with the immunosuppressive environment. The other is the transgenic mouse. So this is the use of an immunocompetent mouse, but in this case, it's been genetically modified to express the human target antigen, so that we can study for on-target off-tumor toxicity. So you can look at that interaction with CD137 and the other co-stimulatory domains. You can look at that interaction with the other immune cells for persistence as well, but it's specifically useful for looking at on-target off-tumor toxicity, because you're looking at the actual antigens that are expressed in humans. And then the use of non-human primates. Now, the non-human primates they have the closest sort of genetic profile to humans. So therefore, there's the advantages of that, but they also have this high variability, genetic variability, as humans do, and therefore trying to get definitive information requires a large number of species of these non-human primate species in order to do the testing. So there's that limitation with that, but it does give us information on, for example, toxicities, specifically ICANS, and ensuring that the human CAR recognizes the native primate antigen for cross-reactivity studies. So it's much more realistic of what would happen in terms of toxicity, because that would be what we would be looking at really here, to what would happen in humans. So this guideline, so this is my overview of the guideline on quality, non-clinical and clinical requirements for investigation of advanced therapy medicinal products in clinical trials. So this guideline was adopted on the 20th of January 2025 by the Committee for Advanced Therapies. So basically, you're going to be looking at the unique characteristics of this ATMP. So this cell-based medicinal products, these product classes have unique characteristics and unique risks associated with them. So, for example, the lentiviral vector has risks associated with regards to the presence of residual lentiviral vectors in your final drug product that could potentially infect other cells because of the tissue tropism of these viruses, even though we want to remove them, and therefore potentially cause problems, other problems. So, but that particular risk is not applicable to induced pluripotent stem cell derived T cells. So you have to take a risk-based approach, and your non-clinical testing has to be based upon the specific risks associated with those particular CAR T cells, or those unique CAR T cells. So, for example, the genetic modification method might produce certain risks associated with off-target editing that may not be immediately obvious, and therefore you need to sort of do long-term studies, if you like. Your non-clinical studies will encompass that. So this risk-based approach, is necessary because all of these products are different. They all have unique characteristics, and they all have different intended therapeutic uses, and therefore you have to tailor your non-clinical studies and the risks associated with that to the product class. Now, your safety testing, so you need to do your toxicology testing and your safety pharmacology testing. Pharmacokinetics studies are required, and the design has to be the studies have to be designed to specifically detect the risks associated with a specific product class, as I've just said. And here are some of the risks associated. I said this in the previous bullet point. So the cytokine release syndrome and ICANS as well, the immune effector cell associated neurotoxicity. So you need to address those risks. What are the biomarkers? What occurs? And you need to have animal models that are based upon these kinds of risks. Now, I want to talk about biodistribution and shedding, because I mentioned it previously, but I didn't talk about it in much detail, and I've been doing a little bit of digging in regard to that. So this is a requirement, So these are done in the relevant animal models. They're often immunocompromised or humanized, and they're necessary to track the vector sequence and the transduced cells outside of the intended site of action. Now, so it's required for your non-clinical studies, and it's also required for your clinical follow-up studies. And basically, you're going to get, one of the problems is that you're going to get residual lentiviral vectors, and those lentiviral vectors are infectious. The envelope, the VSVG envelope, means that they can transduce any, or they can infect any other organs, and you therefore need to design your transgene. I think you would need to design your transgene, build in the quality into the product such that it's only activated or what's the word I'm looking for? Expressed in certain environments, cellular environments, but it's not always possible. But and in addition to that, you've got other issues like if those lentiviral vectors integrate into cells, genomes, and potentially cause insertional mutagenesis in cells that are not related to what we're looking at. So there are risks associated with these lentiviral vectors that have to be addressed. Yeah, so this is where we're talking about the risk-based approach, and that those risks have to be. We have to do we have to look at relevant animal models, and we have to track the vector sequence and the transduced T cells outside of the intended site of action. Yeah. So this is really important, and it's absolutely required for CAR T cells. So it's a mandatory non-clinical requirement, and often we have to do clinical follow-up studies as well to look at this because of the fact that these products are integrating viral vectors, and anything that's integrating viral vectors have a long follow-up associated with them, a 15-year minimum, 15 years. And as I said, the primary safety issue or the safety check is for insertional mutagenesis and therefore insertional oncogenesis. And you know, we will do everything we can to ensure that we have this rigorous washing and purification of your CAR T cells in the final drug product, but you might still get residual cell-free lentiviral vector particles, and those particles would be infused into the patient, and once they are in vivo, they're free to circulate. Yeah, they're free to circulate and potentially transduce other cells, unintended non-targeted cells, such as, for example, hematopoietic stem cells or germline cells. Yeah, and this is the reason why we have to do this. We have to provide tissue tropism data for the same reason, to assess the residual lentiviral vector that could be included in your final product. And I think I did mention it there that, you know, the pseudotype VSVG, that's the envelope, ensures that these lentiviral vectors have a broad tissue tropism, which means that they can transduce other cells, many different types of T cells besides T cells, and therefore you've got to confirm that any residual vector is not transducing untargeted cell population in vivo and is not expressing the CAR protein in an off-site potentially toxic manner. And even if it's not expressing the protein, has it integrated and caused insertional mutagenesis and therefore insertional oncogenesis? You know, so these are the things that you actually have to take into consideration and plan for within your clinical trial. And your non-clinical data, you know, there are limitations to this, but it must confirm that the residual vector is not transducing untargeted cell populations in vivo. So you really, I think, have to look at that VSVG pseudotyping to see if you can just minimize it to just T cells. But I think that it is still infectious. Anyway, so you just got to confirm that the residual vector isn't transducing an untargeted cell population in vivo, and it's not expressing the CAR protein in an off-site, potentially toxic manner. This is from this new guidance, the 20th of January 2025. Well, this is just my interpretation of the requirements of that is required. Again, you need to identify the potential for having an immune response. So in humans, are we going to develop antibodies against the CAR protein, against the CAR T cells? And you need to look at the potential for that, and address it in your non-clinical studies, or at least understand it, because this will affect the persistence of your therapy, of your product, and the safety. You don't want to get immunogenic responses. Yeah, so this is basically what you're looking for in your first in human trials. These are the early phase trials, and the non-clinical data should basically support that. And then your confirmatory trials will be supported by your first in human exploratory trials. Yeah. So your non-clinical data should justify the initial dosing, safe monitoring, as well as administration, and all of those essential criterias. And then for your later confirmatory trials, the data package should be more comprehensive and basic and reflective of an intended marketing authorization submission. But basically, in order to justify your confirmatory trials, you use the data from your information from your exploratory data. So these will be looking at your dosing schedule, your dose escalation schedule, your initial starting dose, your biologically activity, your biological activity data. Right. I don't know. I think the only thing that I wanted to talk about in this slide, because this is a carryover from the previous presentation, is the implementation of the ICH M4Q quality part aspects, and the post-approval change management protocol and annual review requirements post-approval. Everything else is the same. Really, when you're doing your clinical trials and so when you're doing your clinical trial application, as well as your biological license application and your marketing authorization application, it is the totality of information from your non-clinical studies and clinical studies and other relevant comparable clinical trials and clinical data. Yeah, so it is that totality of information that enables not just the regulators, but enables the marketing authorization holder to to make that legal claim, if you like, that this drug can treat these patients. Yeah, so that's really important to note, that it is not just one or two or another, it is the totality of information. And it's particularly the case because you're not able to get all of that information at the time of marketing authorization, and that following post approval, you have your renewal where you will review the data, and based upon the totality of that data, you will then move forward and continue with your pharmacovigilance assessment. So it's really important at every stage of development, of your clinical development. It is the totality of information from whatever studies, whether it be your non-clinical studies, for your clinical studies, whether it be your clinical trials and non-clinical studies and other relevant studies for your license application, and for your post-approval, whether it be your pharmacovigilance data and your clinical data and your non-clinical data and your relevant clinical journals, etc. It is the totality of information that will be reviewed and ongoingly assessed over time. Right, I just want to now talk about the clinical aspects for genetically modified cells, and this is from the EU guidelines on the quality, non-clinical and clinical aspects for medicinal products containing genetically modified cells. Right, the preference according to this guideline is for a randomized control study. Yeah. So you want to have a randomized study and you want to have it controlled by a control group. And the control group is most likely going to be what is the standard clinical practice. And you're making that comparison against this. So you're looking to see what the differences are between your drug or whether or not your drug is superior to or equivalent to what is currently used. And that's the control. As the non-parallel single-arm study can be acceptable, I think you would need to get strong justification and you would need to really get the regulators' agreement, if you like, with that proposal. You need to have strong justification, scientific rationale for that. Um, yes, in the EU, there is this adherence to the principles of the intention to treat. So all of the data needs to be assessed in determining efficacy. So even if patients dropped out or withdrew from the treatment, you need to take their data into consideration. You need to look at all of the data, not just the data, the patients that continued with the treatment, but the patients that dropped out. So this is the intention to treat approach and principles. Yeah, there are just certain considerations that you have to take, clinical consideration from a clinical perspective. These products are complex. You have to have there are specific product characteristics and manufacturing considerations. Um, there are things that are involved. It's not just giving the patient a drug. There's the issues with collecting and handling source materials. Um, there are specific considerations, safety considerations to have to take into consideration with regards to allogeneic as well as autologous origins of cells. So these products are complex and they all have their own specific considerations that you have to account for in your protocoled clinical trials or your clinical studies. And again, because of the limitation with regards to extrapolation of data from your animal studies, yeah, because of the nature of these types of products, you are there's a risk. There are risks associated, and there are processes and there's a guideline that I haven't yet looked at relating to how to conduct clinical trials, first in human clinical trials, to really monitor and to have stringent conditions in place because of the limitations with regards to extrapolation of data. So things like starting dose, biodistribution. So this is going to be covered by follow-up, immunogenicity, immune-mediated toxicity. Again, sometimes it's just not clear from your animal studies until you actually do it in humans that you find the issues. And then on-target and off-target effects. There are long-term consequences of even the on-target effect. It may have potential unforeseen consequences, and then also the potential for tumorigenicity with both of these products, not just the induced pluripotent stem cell derived product class, but also any product that involves an integrating transgene into a host have certain risks related to tumorigenicity. So these are really considerations that are part of this risk benefit approach and assessment that you take when conducting clinical trials. Yeah, again, these are the uncertainties. There's uncertainties with regards to the frequency of adverse events, duration and nature of your side effects. There's uncertainty with regards to persistence in human, and there's uncertainty with regard to immunogenicity. I've already discussed these. These are distinctive features of CAR T cells products that do need to be taken into consideration, and you need to take account for them. More uncertainty. I think I've kind of discussed this, the effects of immunogenicity on long-term safety, as well as uncertainty about repeat dose, particularly with regards to the allogeneic off-the-shelf product. I think I kind of discussed this. Transformation, tumorigenicity, persistence of modified cells, administration procedures. We gain more experience as we go along. We're looking at data that of similar products, but there is still, uncertainty. You need to be thinking about how the other things that you're doing with the patient impacts on the data that you're now analyzing. So your collection procedures, concomitant meditation, and issues relating to lymphodepletion and chemotherapy, how does that impact on your clinical data? And the logistics of doing all of that has to be considered in your clinical trials. Yeah, your dose selection, again, it's based upon your animal studies, but as I said, there are limitations, and there are strategies in place to mitigate the risks associated with administration, starting at the minimum effective dose, or starting at a minimal dose and then doing your dose escalation. And these are the things that we have to review: pharmacokinetics, pharmacodynamics, clinical efficacy, safety, and follow-up. These are important regulatory requirements, as is your pharmacovigilance. So your design has to be appropriate to your clinical design, and your protocol has to be appropriate to the phase. So phase 1 and phase 2 studies are normally done together for these types of products. So you normally do phase 1 looking at the exploratory dose, dose, initial starting dose, and your dose schedules, and your dose regimens, and then looking at the safety first, and then looking at the efficacy. So it's part of the same trial, because patients aren't going to be just used to look at the safety, and that's not it. They want to have some clinical therapeutic benefit. So you have to design your clinical trial for the phase. So for example, phase 3 clinical trial, that needs to be indicative, or it needs to be representative of what how you're intending to market the product. So the choice of your endpoints, trial size, and duration must be scientifically justified based upon the data that we've got. So you can also have clinical data as well. If you're looking at confirmatory studies, you may have clinical data, but how you design your study, your prospectively designed study, has to be appropriate to the to the phase. Your endpoints and efficacy. So there are guidance relating to the endpoint, so your overall survival, progression-free survival, etc. These endpoints are robust, and they must be robust and relevant to the patient population. So yeah, guidance is, yes, there are guidance, EU guidance relating to that. And this is really crucial for these kinds of products. Any gene editing products, whether they be nuclease or nuclease independent, anything that involves integration or genome editing requires long follow-ups of 15 years, is stated in the guidelines, and that's due to these unique risks of, say, for example, insertional mutagenesis and any delayed effects. So there's, it's 15 years usually, from what I've seen in the FDA and in the EU, of consistent monitoring, and you have to put in place financial money in place, funds in place, even if, say, for example, a clinical trial has been ended and you're not progressing, or the product has been taken off the market. You still have to follow up with the patients for that period of time in order to collect that data. So even if you don't go ahead with the data, you still have to do, or you don't go ahead with the product, you still have to do that follow-up, long-term follow-up. Yeah, you have to have a risk management plan, and this has to be detailed in your protocol. So you have to look at the potential risks identified from your non-clinical phase, and determine how they will be monitored in the patients, and that has to be agreed and approved prior to initiation of your your clinical study or your clinical trial. Some of these products are indicated for pediatric treatment of pediatric populations, and I haven't yet looked at that, but I've just got a note here. The clinical investigations for pediatric populations, and I may do a separate presentation on that at a later date, because you want to think about early planning, but in some cases these products are going to be used in the pediatric population, so we do need to consider what the requirements are. Right, I'm just going to look. Oh, I think I was going to look through. I'm not going to do that. That's a mistake. I was going to do a quick review of this guideline on quality and non-clinical clinical requirements for investigational advanced therapy medicinal products, but I've decided not to include that because I think you can look at that yourself. Right, I want to talk about next this ICH-M4. So this is the common technical document for the registration of pharmaceutical products for human use. And this is in a guidance document that's currently draft, but it's really exciting because it's updating the CTD dossier, which has been around for a very long time and does need to be updated to reflect current technology and current advances in medicines. So what has happened and why? Now, so as I say, we're at step 2B for this. It's not currently approved and it's not currently has been adopted. We have had the ICH is still receiving, I think, and in some regions, comments on these updates, but basically it involves a major restructuring and shift in focus of the quality modules. So prior to this, currently the emphasis is on module 3, and module 2 used to be kind of like the expert statement. You know, the expert used to give their opinion on the product, and that used to be what the quality overall summary was about. And then it got turned into this sort of summary of because it is overall summary, it's called a quality overall summary, and it got turned into this summary document that just basically sometimes got updated in post-approval, but sometimes didn't, you know. But now what's happening is they're shifting the emphasis, where the primary document for review is going to be your module 2, the quality overall summary, or module 2.3, and your module 3 is going to be like the repository that contains all the detailed information that backs up the document module 2.3. So there's a shift in the roles of what module 2.3 and what module 3 is doing, and the also the structure, restructuring is really exciting. They're probably going to have some more amendments, but it is really exciting. So what I think is really exciting is the integration of the ICHQ guidelines, particularly on the development strategy and control strategy. So I was going to do a life cycle management manual, but I think that this has kind of covered it, in my opinion. So your overall development and control strategy will include things, for example, like your quality target product profile, your critical quality attributes, and your overall control strategy. And this is going to be something that will be reviewed. I think it's really important. I think it's divided up in, and I'll be giving an overview. I'll be looking at the guidelines and looking at the actual document in this presentation. So it gives core quality information. So it's key critical data points, and the summary and justification, product lifecycle management information. So your lifecycle management, your established conditions, and your post-approval change management protocols, all included in this document. I don't know how it was done before, with regards to post-approval change management protocols. It would have been submitted as part of your regulatory variation submission. But I don't think that your CTD section was updated. I haven't done one for such a long time, so I'm not sure that your CTD section was actually updated. But now that is going to be incorporated, and your change management protocols will be included. So I think it's an improvement, and it just makes everything transparent between the marketing authorization holder and the regulators. And then the other thing is, which makes it really, really good and applicable and bringing it all up to date, is the extended scope and integration. So it's now clearly applicable to biological products and other advanced therapy medicinal products. So it really means that you can add information in specific obvious, clear places where instead of just putting the information in other regions and then having to get guidance from the regulators as to where they will expect to see certain information specifically on starting material, you've got a specific section that is dedicated for the sort of complex information that's required for advanced therapy medicinal products as well as for chemical entities. And as I said, it's aligning with the ICHQ series, and the emphasis of quality by design is been incorporated in this dossier, and the presentation of information therein. Oh, I don't know what happened there. I think I missed the page. Oh well, never mind. So how is it structured? And I will be giving you a video presentation on this, but how is it structured? So it's organized by component type. So you've got your drug substance, you've got your excipients, you've got your starting material. I don't think it's got raw materials, but I can't recall. And it's just a standardized subsection for each component. So for drug substance, you have your standardized subsections. For excipients, standardized subsections, and your starting material. Yeah. And how it's structured is you have your description, you have your manufacture, control, and storage. I don't want to spend too much time on this. So previously, certain information on your starting material, which is required for ATMP, like for example, manufacture, not just listing your starting material, providing your manufacture, providing your quality controls for your starting material, the stability of your starting material, all of that just used to be lumped in your control of materials. It's now treated as with separation and granularity so that it's clear, it's organized in a way that makes it easy to review and easy to what's the word, author and write. Right, so I just want to give a quick overview as to where we are with this draft guidance. So it is a draft guidance, and there are going to be some amendments. Uh, I went through the timetable in that presentation, in that video that I gave. I've got here that the final sign-off is going to be mid-2027, according to the latest work plans. However, that will probably change over time. After this final sign-off, it will be adopted by the regulatory agencies, and you start implementing in the regional areas. Right, so this is the ICH M4Q second revision. So remember that this is a draft document, and I'm going to have a look at the comments to see what they were with regards to the proposal. So it may still change. So if we go to page 14. Let's just move up. So under the drug substance section, so we have this substance intermediate. And for biologics, e.g. antibodies used for antibody drug conjugate or viral vectors used for ex vivo gene modified advanced therapy medicinal products, information should be provided for description, control, and storage. We don't talk about manufacture. And then it says the applicant may provide the manufacturer information separately or integrated into the 233 drug substance manufacture section. Yeah. So the manufacture section can be integrated into that, or it can be provided separately. This section may be used to describe manufacture of specific substance intermediates separately from the main drug substance manufacturing process. So you can do that. This may be relevant for highly complex end-to-end biological drug substance manufacturing processes or cases where subpart of the end-to-end drug substance manufacturing process up to a specific substance intermediate is performed by a different manufacturer. Yeah. So this is the description for substance intermediate manufactured by the same manufacturer and as part of drug substance manufacturing, or it can be integrated. Manufacturing process description should be presented under 233 drug substance manufacture. Yeah. This information should be provided separately in this section only if deemed necessary and contingent should align, and the content should align with this drug substance manufacture section. So basically, if you are going to have a separate manufacture section, then in the substance intermediate, if you are going to have it, then it should also be in this 233 drug substance manufacture section, and they should both align. The information should both align. And just another point, and that is to state that this starting material is the actual genetically modified cells, I believe. Where is it? Substance intermediate. ICH, these are the requirements. So description. This section should include a description of the starting. If starting is sourced biologically, e.g. cell banks or cells used for the manufacture of allogeneic and autologous ATMPs. So that's the initial cellular starting material and the cell banks as well. So for biological starting material, information on the procedures used to generate working cell banks, seed lots, and or cell modification procedures should be provided if applicable. Generation of master cell banks should be described in the 234 starting material. Yeah, so 234 starting materials. Let's have a look at that. So the 234 development and summary justification. Here it is. So this is the description. So here, for biological starting materials, this section should include information on the establishment of the master cell bank, working cell bank, or the virus seed bank systems. And these are the relevant information with regards to what you need to provide, as well as your manufacturing process. And then in the modules, the review section, so this is the module, the old module 3, all the quality documents. If we go, I don't know whether it's not gonna, if we go to this part, drug in the drug substance section, so we go to 35, let's see if we can, this is still module 2, so module 3, so the drug substance manufacture, viral clearance studies is now integrated into this, which makes sense. Well, to me it does anyway. Substance intermediate. So this is the control. This is this something different. Manufacture, additional information on manufacturing, starting or materials may be provided as appropriate. Yeah. So you just have to be very careful, I think, when you're putting together these dossiers to make sure that the information goes in the right section. Yeah, I think I might put together a template, a checklist, and put that on my website with regards to when you're preparing each document, what goes into what sections. So this part of the presentation is on the ICH-M4, and as I said previously in the previous slides, because I kind of integrated this presentation into that original presentation, so it might be a repeat, but the basic in terms of the control of materials, or critical starting materials, this shifts from the S23 to a separate substance intermediate. Now, this is a draft guideline at the moment. So the draft ICH M4Q guideline introduces the concept of a substance intermediate. And in the section 2.3.3 SI, yeah, and the way that the presentation of the dossier is going to be is that the shift is going to be from, as I said in a previous slide, from the module 3 to module 2.3, and module 2.3 will be the main review and focus of review, and module 3 will be the repository. Yeah, so S233 SI is the place where you will have the substance intermediate to better accommodate complex starting materials and complex components of biological products. Yeah, so in this section, it states that the viral vectors used for ex vivo gene modified advanced therapy initial products, right? So it should be addressed under this new 2.3.3 SI substance intermediate section. And the rationale is that the new classification acknowledges that lentiviral vectors is highly manufactured, it's highly controlled, it's manufactured and highly controlled, and it requires extensive documentation, just like as a drug substance. But it is used in process to create a final active substance, active ingredients, which is the CAR T cell drug substance, and therefore making it an intermediate step rather than the raw material or the final active itself. Well, you can't make it the final active substance. Well, you could if it was going to go in vivo, but it isn't. This is an ex vivo transduction of cells, yeah, to make genetically modified gene cells. So this is the way that the ICH guidance is going. But remember that this is a draft guidance. I think it's a good idea. I think it makes sense to me to separate it out. But let's see what gets approved. This is my references. Some of the key references I've listed here. They're critical for understanding these regulatory landscapes, I think. Please feel free to refer to these materials at your convenience if you want. Then I don't think I put all of them because I've got a lot of slides on references, and I don't want to. Here are the relevant US guidelines. I haven't put the legal framework actually written that down. I might have referred to them. I don't think I have actually. But here are the guidance documents for the FDA. The ICH documents that I've referred to, or I've talked about some of the contents of which. More ICH guidance documents. And here are the EU legal and regulatory framework that I refer to. I think there's a lot more. I didn't put another one in there, another two in there, but I referred to it in the presentation. Well, that's the end of my presentation. It was quite a long one. I hope you enjoyed it. If you've got any comments, please don't hesitate to ask. I hope to see you at my next presentation, which will be Milestone 3, and I'll be talking about the quality aspects. Thank you so much, and goodbye.